Hi everyone, welcome back. So we are going to start now our final panel and final session. We'll explore how genomic has been applied across the healthcare spectrum to personalize care. And that is whether you're choosing the right medication or improving cancer treatment or enhancing outcomes for transplant patients. Guiding this discussion is Dr. Sterling Bryan. Welcome. Sterling is a health economist and professor at UBC School of Population and Public Health. He's a senior scientist at the Center for Clinical Epidemiology and Evaluation and currently serves as the Chief Scientific Officer of the Michael Smith Health Research BC. Dr. Bryan's research focuses on the cost-effectiveness of genomic interventions, including in depression and kidney transplantation. He is also president of the Canadian Association for Health Services and Policy Research and a fellow of the Canadian Academy of Health Sciences. So please join me in welcoming Dr. Sterling Bryan and his panel.
Great. Thank you very much, Federica, and thank you for the invitation to moderate the final panel today. I just wanted to start off by acknowledging that Michael Smith Health Research BC is a partner organization with Genome BC, and we're delighted to have the opportunity to support today's event. So thank you for joining us for this session. It's called Genomics and Precision Medicine, Personalizing Care Across the Spectrum. So what we're going to do in this session is explore how genomics is driving personalized approaches to patient care, from tailoring medications through pharmacogenomics to advancing cancer treatments, improving transplant outcomes, and also understanding a patient partner perspective. So I'm delighted that we have a patient partner joining us for this panel. So we have an excellent group of panelists, and I just wanted to note some of the guidance that I had from Genome BC in moderating the panel, one of which was, do not be afraid to cut off somebody who is rambling. So if we have anyone rambling, then I will attempt to follow the instructions. We have an excellent group of panelists. So we're first of all going to hear from Dr. Christian Steidl. Dr. Steidl is the research director of the Center for Lymphoid Cancer and executive director research at BC Cancer, and a professor in the Department of Pathology and Laboratory Medicine at the University of British Columbia. We'll then hear from Dr. James Lan. James is an assistant prof and clinical scientist jointly appointed to the Department of Pathology and Laboratory Medicine and the Division of Nephrology at UBC, and is the medical director of the Vancouver Immunology Laboratory and a transplant nephrologist at Vancouver General Hospital. We will then hear from Lisa Ridgway. Lisa and I know each other well, and delighted that we have Lisa as a panelist. Lisa is a patient partner and formerly a lawyer. She partners with UBC, with UVic, Island Health, and the BC Support Unit at Michael Smith Health Research BC, and advocates for lived experience perspectives in health research. And then to close the presentations, at least, we'll hear online from Dr. Bruce Carlton, who's not able to join us in person, but will be joining us through Zoom. So Dr. Carlton is a professor at the Department of Pediatrics in the Faculty of Medicine at UBC and investigator at BC Children's Hospital. So in addition, Dr. Carlton leads the Translational Therapeutics Division at UBC and the Canadian Pharmacogenomics Network for Drug Safety. So at that point, what I'm going to do is make sure that we have this as a conversation. So you have online, you'll have the Slido Q&A, and we'll make sure that there's time for questions at the end. So please feel free to add your questions at any time, and we'll get questions from in the room. But first, we're going to have a poll question. So if we could put the poll question up, please. So if you want to log into Slido and tell us which area of precision medicine do you believe will benefit most from genomics over the next 5 years? So which area of precision medicine do you believe will benefit most from genomics over the next 5 years? So I'll ask you to give us a response to that poll question, and then we will come back to the answers towards the end of the session. So we'll now move to our short presentations, and we're going to start with Dr. Christian Steidl, please. Thanks.
Okay. Hello, everyone, and thanks for the invitation. And Sterling, I take your threat very seriously, both on the time and the rambling. So before I start, I want to convey my gratitude to being invited, and thanks for Genome BC for having me here. But I'm actually double grateful because some of you might have seen me, limping around in the venue, and so actually I had a ski accident. And if you hear me cough or be out of breath, then the reason is that I'm probably giving a talk for the first time in my life with a pneumothorax. So I hope it all goes well. But I'm feeling okay. So with that. The topic of my talk is essentially verbatim, as you see on this slide, what the title of an LSARP was that the lymphoma group conducted together with their funders, Genome BC, Genome Canada, PHSA, CHR, and the BC Cancer Foundation. So is this the buttons? So obviously, this type of research is massive scale and involves many colleagues and supporters. So you see here the team members, some of which are in the room, a broad collaborative effort between the Center for Lymphoid Cancer at BC Cancer, the Genome Sciences Center, and also a very healthy representation of health economists in this team, and with a view on the Genome Canada, Genome BC processes. I also put the research oversight committee in here, and you know that it can be a love-hate relationship. So we had a very good one. And so I just want to give a shout out to an excellent chair. And I think this implementation was really how it was supposed to be, that it was a productive relationship with lots of input that could be onboarded. So to go back to the origins, in 2016, when we conceived of this project focused on lymphoid cancers and specifically the problem of relapse. So just want to acknowledge here that I believe everyone can find the spin on their own project, why it's the most important in the world. Let me try that here for lymphoid cancers and give you my own version of the cancer statistics here to show to you that from a perspective of cell of origin and a cancer derivation from lymphocytes, that you can tally up all these cancer types to 7.7% of all cancers, which would then rank lymphoma or lymphoid cancers as quite common at place number 4 or so. And specifically in 2016, we realized that the clinical problem of relapse should be in the forefront. That's where mortality is driven, as you can see in the outcome curves on this slide. You see the scale in terms of how many patients would be affected per year in Canada, and also a health economics bill represented on the slide. And needless to say that all of this amounts to significant impact on families and society at large. And if you look at the outcome curve here in the middle of the slide, there is the notion that lymphoid cancers are very treatable and with very high survival rates. And that is true, but only to the point where first-line therapies are effective, and then curves drop quite significantly to what you're seeing here, with only a very small residual sliver of survivors when first relapse has occurred. So this is now a bit mitigated by modern immunotherapy and targeted treatments, but still it holds very much true as one of the big unsolved problems in lymphoid cancer care. So then in our LSARP project, we configured our projects along this virtuous circle where we wanted to deliver two things. We want to keep discovery really as effective as possible with onboarding of new technologies. You see that on the left-hand side, but at the same time also distill the relevant information from the Genomics Foundation and develop very concretely biomarker assays in the publicly funded healthcare system. And you see a selection here. I will in the interest of time, only just give you short vignettes of how we have actually delivered that during the tenure of our funding. So this virtuous circle idea is really very much alive in the program that in the lymphoma program as a whole, but also was, I think, quite effectively executed in the LSARP. So what underlies the structure and the infrastructure of the Center for Lymphoid Cancer is really the idea of having a population-based approach to acquisition of patient samples, but also data. You see here conceptually how materials from our patients that are fully consented and also data acquisition flows through the system and can conceptually be separated into the database and a biobank. So one quick remark on the biobank. There is this corner in the bottom there that we call the virtual biobank. So this is mostly driven by formalin-fixed paraffin-embedded archival tissues that we have an effective system in the province to get access to these specimens. And even though they are not permanently in our hands, we just have an effective system to interface, thus virtual. But most of our genomic studies these days are actually built around the availability of FFPE tissues, which obviously enables very statistically powered study designs based on the numbers. So these are the project numbers in our LSARP. Really briefly to give you an idea of the scale, you see many numbers in the hundreds across the most common lymphoma and leukemia entities shown here. And one thing to highlight is that many of those samples are sequential biopsies. So with the idea of capturing relapse-specific biology, it's very important to have from the same patient multiple biopsies that we can study, and that was a unique strength of our study designs. So now, as I said, it's impossible in 10 minutes, and I'm seeing that I'm only having 2:40 left here. So we have to quickly go to the essence of what we believe biomarker assays should look like. If we do a distillation from the broader genomes and transcriptomes that have been produced in the project. And these biomarker assays are, in essence, classification assays. They are supposed to improve nomenclature and subtyping of lymphomas. And you see here how this can be done as a biomarker assay to build a spectrum of diseases that are distinct, and how a modular design of our here a gene expression assay can distinguish molecular entities within a parental entity, and shown here diffuse large B-cell lymphoma, so-called primary mediastinal large B-cell lymphoma, and dark zone signature lymphomas. And all of this information has personal utility to patients. It has clinical impact in treatment selection and also titering treatment intensity. So this is now the implementation that we put forward as a major deliverable from our LSARP. So we have a test that is available to all British Columbians as we speak. So this was routinely implemented in pathology at BC Cancer in October 2023. So we have a throughput of roughly 5 to 10 samples per week. And you see that this curve is steadily inclining. So this is now available to our patients, and it's clinically useful, and we take this forward as a paradigm of how now future assays that come from the discovery can also be implemented. And we are, of course, going to such discussions in our panel discussions, I'm sure. So I want to end with a quick outlook what is next on the horizon. And I just want to throw out that concept. I think Marco has alluded to this in the morning as well, that spatial information through genomics can be layered onto the more traditional genomics approaches. And here, a biomarker study that we have published in the Journal of Clinical Oncology that emphasizes this concept that spatial information makes our biomarkers more accurate, more clinically useful. And in this implementation, we have a prognostic biomarker that's risk stratifies Hodgkin lymphoma patients quite effectively. So our unique contributions were that we worked with relapse biopsies. I mentioned the formalin-fixed paraffin-embedded tissues as a viable strategy. Tumor microenvironment with the spatial topics is a new frontier. And I didn't mention so much the health economics study that was performed alongside the entire ELSA project. I think that helped with implementation. And these were the questions that I came up with that could be addressed in the discussion, in the panel. We can talk about clinical utility and oftentimes inadequate biopsy specimens in pathology labs. I stop here because I'm seeing that you're getting a bit nervous on my left here. And with that,
great. Thank you very much.
You can take over.
Great. Thank you very much indeed, Christian. Just wanted to clarify one point. You said the health economics bill was 315 million. And just to be clear, that was not the funding that went to the health economists in the team. It was the cost associated with lymphoid cancer. Okay, excellent. Great. We'll move straight on. James, please. We'll move to transplantation. Thank you.
Thank you very much. I'm honored to be here. I don't have a pneumothorax, but I'll try to stay on time as well. Here's my disclosure slide, which is not present for the information I'll present today. So I know not everybody here wakes up thinking about kidney disease and transplant. So by way of background, kidney disease occurs in about 1 to 10 Canadians. And for those that have end-stage kidney disease, the mortality is extremely high when you have to go on dialysis. We think about mortality for cancer patients, but for those on dialysis, it's 50% at 5 years. And for those number of patients, they cost the healthcare system billions of dollars every year. Unique to kidney transplants, one of few therapies I know that will improve patient quality of life, save their lives, and also save the healthcare system dollars. And I think we're very proud in BC because we're the only place that I know in North America where the number of kidney patients on dialysis, there are more of them treated with a transplant than with dialysis at this point in time in BC. But what is the problem we're trying to solve here? And that is rejection. Half of the transplants will fail in 10 years. And the dominant reason is antibody-mediated rejection. So the issue is that the immunosuppressants that we give, they're not enough to attenuate the immune response. And over time, patients develop T-cell or antibody-mediated response causing rejection. So difficult is a problem that we don't actually have an effective therapy for patients encountering rejection. And so when LSAP started, we were lucky enough to get this grant to establish what we call the Genome Canada Transplant Consortium. This is a unique effort that ties together over 100 scientists, many across the world, and also we have involved regulatory bodies, health economists, as well as law and legal, so that we can tackle this problem together. So what was our hypothesis? And we kind of went back in time, and we remember that our first transplant occurred between identical twins, because back in the days, there was no immunosuppressant. And that transplant could be undertaken because the donor was not viewed as foreign to the cell. And we hypothesized that there are these immune molecules in our world, the human leukocyte antigens, that there are these discrete epitopes on their surface that incite the immune response. And now we hypothesized that the more of this disparity in amino acids, these epitope residues on the donor compared to the patient, that would indicate a high risk for rejection. So we kind of set out to find out, well, is that going to be the case or not? So the first question we answered was, can we use molecular mismatch analysis to identify patients at risk of immune rejection? And we wrapped up this analysis recently. This paper is under review. In BC, we analyzed 1,000 patients transplanted recently. And what we found was that there are indeed patients with a low number of molecular mismatches at these epitopes on the surface of HLA antigens. And those patients are at very low risk of rejection in comparison to those with high mismatches. And so that was very intriguing to us. The follow-up question was then, can we use that information to guide the type of immunosuppressive medications we give to our patients? And what you're seeing here on the left is that patients with a low number of molecular mismatches, these epitope differences, it didn't really matter to them what type of immunosuppressive therapy that they got. They could receive very minimal immunosuppressant or very intense immunosuppressant, and they had very low risk of rejection. On the right, what you're seeing is that the patients who were not so lucky, they had a high burden of mismatches with their donors, that there was a high risk of rejection if their immunosuppressive therapy was not intense enough. And so for those patients, the lever of immunosuppressant would be very helpful. When we modeled this out, we wanted to understand how much can we reduce the impact of rejection in our cohort. And we found out by knowing this information, we could reduce the type of rejection to less than 3% that we see clinically by 1 year. When we did this research, people kind of asked, well, are you just presenting retrospective studies, or can you actually go ahead and implement this? And one of the key components for us was that we wanted to see, can we move this idea into the clinical arena fairly quickly? How would that look like? So what we would do is then we would sequence all of our patients, identify their genotypes awaiting for transplant. And whenever a donor would come forward, we would do the same for them as well. We would do that using high-resolution typing technologies, and then convert the sequence into a 3D protein antigen structure to compute their epitope differences. And depending on the degree of mismatch, then we can guide the prescription of immunosuppressant. Unique to transplant, I think we are working under time pressure all the time, because when a deceased donor passes away and donated an organ, we only have a few hours to really identify the difference between the donor and the patient and the degree of their mismatch. And as you guys know, most of the technologies we have today are unable to do that, such as the MySeq instrument that you see here. That would take days to get the results back. So as part of our LSARP, we had to advance the technology that can give us real-time information. Unfortunately, when we started LSARP, that's when the nanopore came onto the scene, which we thought was excellent because of the ability to get real-time sequence information that is suited for deceased donor transplantation. So we were working with a company, and we completed this validation last year and demonstrated that across all types of DNA specimen that we have buccal DNA, very low concentration, we're able to get 100% concordance. And we were able to get the results within 4 hours. This is one of our postdocs who did the experiment in real time as deceased donors came up, and we were able to get the results that we needed in a timely fashion. So how does the workflow look like in clinical practice? Cue the video, please. So this is what happens now when the donor comes up. Our technologist gets the blood from the donor, gets the DNA, sets up the PCR, and a very easy library prep method. Then we load it into the flow cell. We have in our lab the MinION and the GridION as well too. The sequencing time when we started was an hour, but that's now shortened to about 30 minutes because we found out we have enough reads with 30 minutes of sequencing time. And then we do the analysis, that's only about 10 to 15 minutes to get a genotype, and we convert it into the 3D structure for the epitope analysis. And so for this patient, what you're seeing here is our technologist putting into our pipeline to get the derivation. And so from beginning to the end, we have that data completed in 4 hours. We're also setting this up as a serial sequential kind of way to sequence our specimens in the lab as well too. We are also a clinical facility that does typing for BMT and solid organ transplantation. And so this technology is very scalable as well. And so really that technology now allows us to get all the data that we need when a donor presents themselves so we can prescribe the type of immunosuppressant that we want. But then we quickly realize that if we leave it up to chance, the majority of patients are not going to be well matched with their donor. So what that effectively means is that most of the patients, they have a mismatched donor. They're not very well matched. And their safety zone for over and under immunosuppression is quite narrow. And in order to fix that problem, we have to really go back to the top. When we allocate the organ, can we allocate every organ into a patient that they're well matched so they don't need so much immunosuppressant? So that's the second part of our story. And we're lucky to get the GAP funding to now prospectively apply epitope matching strategy for renal transplantation across the country. And you can see the number of institutions and partners that we have to help us with this work. And one of the concerns that first came up was, well, are you sure that for every donor, you can find someone well matched to give that kidney into? And so we conducted this modeling exercise across Canada. And what we found was that indeed, if we actually want to find every an organ, a well-matched donor, that is very possible, even when the waiting size is fairly small, as you see on the left side, less than 100 patients waiting for a kidney transplant. The second question that was brought up by our patient partner team was, well, do you worry about ethnic disparity? Is there a concern that if you're an Asian person living in a predominantly Caucasian society, that you're going to be more difficult to find a well-matched organ donor? And Dr. Doyle, who worked with me on this data, was able to perform that analysis and found out that actually, as a matter of fact, ethnicity, at least in BC, did not determine which candidates could find a well-matched organ versus others. So in conclusion, by the end of the LSARP, we were able to really develop a real-time application of the genomic technology to precisely guide the selection of immunosuppressive medications. We're now able to incorporate this new technology and the framework of precision medicine into organ allocation. So there's a link into policy implication as well, so that we have the potential to minimize rejection. And I think this is all the foundational groundwork that we hope to advance. The next holy grail in transplantation is if we can match organs into patients, they're all well matched, can we minimize drugs and achieve tolerance, which is that patients don't need drugs at all. So thank you very much for your attention. I want to just acknowledge the many, many collaborators and institutions that are together, working together on this. I'm just someone presenting the work on their behalf. And thank the Genome BC and Genome Canada for LSARP, GAP, and now the healthy outcome through genomic innovations. Thank you.
Great. Thank you very much indeed, James. So I'll ask Lisa Ridgway to come up. One of the things so Lisa is a patient partner and formerly a lawyer. So one of the things I love about Lisa is that she refers to me sometimes as her learned friend. So I'm going to ask my learned friend to come and present on behalf of patient partners. So please join us, Lisa. Thanks.
Better adjust the microphone. It is brilliant to be here today. Thank you, Genome BC. Thank you, Dr. Julia Hill, Dr. Chenhua. I'm Lisa Ridgway, a patient partner in research, and I live and work on the unceded lands of the Lekwungen-speaking peoples on Vancouver Island, and it's a beautiful place, and I'm very grateful to call it home. So standing here, I'm reminded, perhaps Sterling knows, the 1971 Monty Python sketch, and now for something completely different. Are there any patient partners in the room? No. Okay, are there any lawyers in the room? No. Okay. Well, I'm sure there are some of you virtual, so that's good. I'm going to start off by telling you what I'm not. I'm not a geneticist. I'm not a health scientist. I'm not an economist, but I do not represent all patient partners in research. I'm going to try and represent my team, but ultimately it's just me. I am a recovering lawyer, and most importantly, a patient partner in research on a project that Genome BC has funded for 6 years. Yes, I got the invite in March 2020 and never looked back. So next slide, please. Speaking of economics, here's my dog, Dazzle. One of my colleagues said, please bring something to the team and the presentation about yourself. So this is my dog, Dazzle. And if you look carefully, you'll see that she's doing a literature search in The Economist magazine. And if you look really carefully under the chair on the left, there's a certain orange guy on the cover. What a good dog. So why am I here with these distinguished gentlemen and talking to this very learned crowd? I'm here because patient partners belong in precision medicine research as much as people in white coats or with suits and ties or with calculators or electron microscopes. It really is the so-called golden age of precision medicine. I read that the other day. I thought it was a great phrase, the golden age. So there's lots of evidence and fact in my work as a patient partner and just important funding. And that dovetails completely well with my heartfelt opinion that our place, the place of patient partners, is right here beside you. So there are 2 projects, next slide please, that I'm involved in, 2 phases, I guess, of the same project, mental health care and pharmacogenomics. In 2023, our team established that PGX testing could save the BC healthcare system close to $1 billion. We're in the implementation stage of our project. We've got 1 year to go. It's going to be a big year. I know it's customary to thank your team at the end, but as usual, I'm doing things a little differently. Sorry, Sterling. Many people here helped ensure my success. Our project manager, Dr. Louisa Edwards, our team's knowledge translation guru, Dr. Alison Hoynes, our world-class geneticist, Dr. Chad Bousman, I call him the gene sequencer, my fellow patient partner and co-author, Linda Riches, our modeling genius, Dr. Shazad Ganbranian, the students and the trainees, especially in genetic counseling, that are the future of our work and who will carry on well past my best by date. Congratulations, Mary, wherever you are online, I hope, on your defense. And last but very not least, Dr. Janine Austin, Dr. Sterling Bryan. Both of these people have taught me climbing skills. And climbing skills means to climb up, but they've also taught me how to climb down, including today. So what moves health science forward? Next slide, please. Proleptic thinking, the ability to join ideas, see patterns, ask questions, and drive forward with an absolute trust in research methodology and empirical evidence. So my idea is to talk about 3 themes that have changed actually not an iota since the project started in 2020. The first theme is teamwork. That's the real power behind our success. The second theme is persuasion, and I'm going to try and persuade you about patient engagement and patient-oriented research. And the third theme is persistence, the don't let the blankety blank get you down. Now you can please fill in the blank, whatever blank you want to fill in. Anyways, this is not about me. I have a great story. Some of you know it. It's been published internationally. One thing I do hold a dedicated position in the Department of Psychiatry as a patient partner at UBC. I've been there for 6 years. Thank you. You're not here, but Dr. Fidel Villa Rodriguez, I'm his co-PI on a randomized control trial and a number of other projects. Our latest gig, it's really cool. It's creating plain language summaries and infographics to 100 published papers. some of them are mine, to build up patients' knowledge of treatments for depression. That's a paper a day. We're summarizing a paper a day for a year. It's really going to up my stats on PubMed and ResearchGate. So I'm never going to be a member of the Royal Academy of Science in Canada. Is that a thing still? It is, yes. But more about that later. What I'm going to try and do is convince you to seek out patient partners, engage in your community, and welcome quirky voices like my own that may not exactly understand what you do, but we really do share your goals. So let's talk about reaching goals. I presented with James. It was a good day a few months ago, and a colleague in the audience asked me whether or not it was a conflict of interest for a patient partner to serve on a team that might in the end benefit or help their own condition. So sort of like, are you self-serving, as it were? And I thought long and hard about that question, and I should have responded with, well, sir, what drives you? I mean, is it self-interest, or is it the interest in trying to help, to change, to improve the human health condition? Because that's what drives the very dedicated patient partners that I know. And I just wish I was smart enough and fast enough at the time to answer that question, but I've just answered it now. So teamwork, persuasion, persistence, the plow horse power of teams pulling together. Not just a bunch of race horses trying to outrun each other or the clock or the funding. This is a really good team. And sometimes we don't agree. But Dr. Edwards, thank you. You helped us craft guiding principles of working together, and those principles have been updated a lot, and they've withstood the test of 5 years. So next slide, please. Here's my theory. I just thought of it on the weekend. Take your meds. Model engagement, educate patient partners, embed in your community, demonstrate what's possible, and standardize ways of working. My drug of choice, the med that I take, is patient engagement. Our project has a patient partner on our research oversight committee sitting at the head of the table. It's wonderful. Thank you, Genome BC, for that. There really is value in patient engagement. We add experiential value. We, in my case, full disclosure, taken the drugs, done the genetic test, which, by the way, didn't work for me, suffered through some stigma. So patient partners actually add something that you can't measure. We add real life. So no one on our team or viewing our team from the outside is ever going to say that we did the research in some quiet, hallowed hall or dreaming spire or ivory tower. So that's the persuasion part. I'm going to persuade you with some passion, but I'm going to give you some hard numbers too. Next slide, please. The BC Support Unit is a federal strategy. It's been around for about 10 years. These are some figures and facts that they have collated. The BC Support Unit operates with the kindness of the Michael Smith Health Research Foundation, also with Clinical Trials BC and Research Ethics. So persistence is my last theme. I'm almost there, Sterling. I've got 11 seconds. I've known rejection and failure professionally and personally. I've done 2 bone marrow transplants with my brother. One worked, one did not. I still can't find consolation in that he got extra years perspective. That was rejection. So then there's failure. Failed grant applications, failed patient partnership. I mean, we're only human. One thing really stings, though, if we, in this room and out, are to accept lived experience as an indisputable form of evidence and tell our stories, those stories are facts. And how we tell them matters. So if a major journal calls my first-person experience an unreferenced, ill-thought-out piece and rejects it summarily. That happened not too long ago. It not only rejects me and my co-authors and my team, it rejects patient experience. I think I'm over the shock of that rejection, so there are just the lessons to be learned now. Because whether you like it or not, or accept it at this point in your research project, patient partnership in health research is coming. I mean, it's here, right here. And there are others, patient partners behind me, climbing the podium, climbing onto the platform, putting ladders up against old science and trusting each other to reach down and pull up other patient partners. So don't think of us as individuals. I mean, we are, but please think of us as like a tide, because we need you to help harness the motion. Patient partners in research, I don't know if you know this, but we talk to each other all the time about methodology and best practice, about science, about health research, and we celebrate each other's success. I mean, just recently, a patient partner was promoted to the board of Canada's drug agency. So we're quite determined. Anyways, that's my story. It's all about trust. I think I have one more slide. Yeah, it's all about trust. I just really believe in what we're doing on our team. I believe in patient-oriented research. I believe in what we're about to do. My lived experience is like a passport to health research, and I'm crossing some borders, and I've probably crossed some boundaries here today. Thank you, Genome BC, and thank you all of you for your helping hand.
Thank you so much, Lisa. That's, I think, a great statement and something that I truly believe is that lived experience and patient partners change everything in the way that research happens and change things in a way that is going to enhance and raise up the work that we do. So thank you for that, indeed. Something that I'm learning from this is that I never want to be following Lisa Ridgway when I'm giving my presentation, but Bruce, I'm afraid you're going to have to follow Lisa, and I'm sure you'll do a great job. We look forward to hearing your presentation, Bruce. Thank you for joining us. The floor is yours, Bruce.
Okay, thank you. And welcome, everyone, and thanks for inviting me. I'm sorry that I'm not there in person. I was at the American Academy of Pediatrics meeting and just got off a plane, so I'm glad that I'm able to speak to you today anyway about the work we've been doing in precision medicine, and particularly in the safety of drugs in pediatric cancer. The overarching goals of our precision medicine network, called the Canadian Pharmacogenomics Network for Drug Safety, or CPNDS, is to make the lives of children better. We want them to survive, of course, their cancer, but also thrive. And we can do that by optimizing drug therapy to achieve its best outcomes possible for every patient. We're not necessarily as interested in our network in newer drugs, creating newer opportunities for survival. Survival is very high in pediatric cancer. At 5 years, almost no patient ever has a relapse. And so we do talk about cure in pediatric cancer. And at 5 years, more than 83% of children with any type of cancer in British Columbia survive and are cured. But one of the key problems that we still have is harmful outcomes from medication use. So we began working on testing patients before chemotherapy to understand their risk of adverse effects. There was quite a bit of resistance from the oncology community at the beginning, uncertainty about how the information could be used, and worry that providing such information about harm would make families more resistant to using drug therapy for their children's cancer. That didn't turn out to be the truth, but that was certainly an early concern. From the early days of the project till the end, we saw almost 1,000% increase in requests for testing of the project. And pediatric cancer is very rare. In British Columbia, we have between 120 and 140 new cases of pediatric cancer a year. And so to test 1,677 patients with pediatric cancer across the country in this project is actually quite an accomplishment. We accomplished testing in about 60% of all children with cancer. So we don't test everyone, not yet, but we're still working on that. And we get a lot of requests for new testing to be done in other areas besides pediatric cancer. Some of our major achievements were collaborating with and getting the recognition of the work that we're doing with the Children's Oncology Group, which is the world's largest children's cancer treatment agency that standardizes treatment and it has, as such, has resulted in the great survival rates that I quoted earlier. And so we're now working with their germ cell committee, their soft tissue sarcoma committee, their rhabdomyosarcoma committee, and we've put in several applications for funding with them on behalf of work to improve the safe use of drugs in children. Along with that work, we've also discovered 26 new genetic variants that indicate risk of or protection against 5 specific serious and severe adverse drug reactions in pediatric cancer. And in the process of doing all of this, we've created probably the world's largest cohort of pediatric patients with genomic and comprehensive drug outcome data in the world. And this resource is part of our new strategy, will be to expand and make this available so even more genomic discoveries can be made to improve the safe and optimal use of drugs in children over time. At the International Pediatric Oncology meeting in last October in Hawaii, there were 3 presentations from our group, 2 of which received Young Investigator Awards, all about the work that we've been doing. This follows a series of years in which there were no presentations on pharmacogenomics in pediatric cancer. So we're pretty excited about the fact that we were able to achieve 3 presentation slots in that international program to talk about the work that we've been doing. So we've seen international and national recognition for this work that was funded by Genome Canada and, of course, Genome British Columbia, our great partner. And we've created a model and infrastructure for national implementation of pharmacogenetic testing. We test kids across the country from Vancouver to Halifax, and we've seen both social and economic benefits of testing in many ways that we can discuss in the question period if you'd like. Our future directions are to do more cell-based and animal model validation of newly discovered genetic variants. We can talk about that further too if you'd like. We are expanding our pediatric oncology test panels now in a new project, similar to James's presentation on the Hoggy project. We're also doing a project with Genome Alberta in which we're expanding our test panel. We're moving from research-based testing to clinical testing in the lab to make the testing more widely available and cheaper for patients over time. We're doing some computer-aided drug design, modeling specific targets to determine the best treatment strategies to activate or suppress these particular targets to improve drug safety. We're doing both preclinical and clinical interventional trials now based on data that we've published in this time. And we're leveraging our data bank and improving it also with funding from Genome British Columbia and Genome Canada to support increased bioinformatics research. This is all going to be online data. The data will be is all processed on very powered servers. And all the analytical software you could use to work with next generation sequencing data will also be online and available. So the data won't just be able to be downloaded for analysis. You can actually do the analysis right online using your laptop, because the servers in the background can accomplish the directives that you send from your laptop. So we're really excited about that. In the next 2 to 4 years, we're implementing that now. We're just getting that up and running, and we'll be educating people on how to use this to increase the global use of our data, to improve medication use and optimized use in children around the world. And finally, we're working on affordable pharmacogenetic testing for patients that will be available in pharmacies in several provinces. And when I say affordable, I mean $40 tests, which is quite an undertaking to make these inexpensive but yet accessible to more patients, to move the process forward. And my last slide, I just wanted to say that this is something that we're particularly interested in, and we call it the Extraordinary Responders Project. In many ways, we've been told to look, usually by well-meaning and appropriately by biostatisticians, to understand the power of a sample to answer specific questions. And so you look at average effects over time. The average response, for example, the peak of the bell-shaped curve. That's what most randomized trials tell us about, the average response. But we're particularly interested in the tails. We're interested in the super responders, or what we call the extraordinary responders, the patients that either have a miraculous response or have serious harm, or a combination of those, and what makes them different, and how can we exploit that understanding of those things to give more people a miraculous response when that hasn't happened in the past. Why is it genetically that they're not responding as well as others? Why is it genetically that someone has an adverse response and others do not? And this is the work that we're currently doing. And I'll stop there. Thank you.
Great, thanks very much, Bruce. So we will now continue with the conversation. So I'm going to start by I try and be a glass half full type person, but I'm going to pretend to be the glass half empty guy to start with. So I'm going to ask you to reflect on barriers to bringing genomic insights into treatment in clinical practice. And what are the sort of strategies that we should be thinking about to ensure that we see these genomic technologies and the genomic advances being implemented and used in clinical practice? So I think, James, can we start with you? You've actually got a success story, I think. So how can we all be like James?
Well, I'm not sure that we're quite there yet. When we started, we really want to make it a practical tool that we can use clinically. And we thought the first barrier was going to be technology. And in fact, it turned out to be the easiest one to address. That when we started in 2015, 2016, they thought we were crazy to use Oxford nanopore because the technology wasn't accurate enough. But through better accuracy and chemistry, we were able to kind of get there. And then we had to make a case for implementation of technology with the health authority. And that part wasn't difficult to do because it turns out the costing of this technology is comparable or better than the ones that we use. And once we crossed that road, it became, well, if we can do it, use the technology to inform risk stratification, would the clinicians act on the information that we have? And there we had to include the input of clinicians on how they would actually use that data, and how can we validate what's the anticipated results. And so you can only do so much with retrospective data analyses and have some anticipated benefits that you expect. But we also want to make sure we're not causing harm by switching to a new approach. So when we switch over to the nanopore-derived risk stratification and then given medications based on that data, then what we did is then we have to have a way to verify that the rejection rates are actually going down. So we had to talk to the pathology team to make sure we have real-time data collection on the rejection rates to analyze them, and that we're actually getting the anticipated benefits. So that part has been okay, but the most challenging part actually has been the intersection with policy of organ allocation. Prescribing medication based on precise risk stratification information, that part, I think everybody can buy into it. But when we discuss that, we're gonna actually give a kidney to someone who is well-matched. That part has policy implication, and that's the most challenging part that we have to tackle, because all of a sudden we're dealing with a precious resource. And instead of giving to someone who has waited for a long time for that precious organ, we're now saying give it to someone who is a better matched patient that may or may not have waited the same amount of time. And so I think that part with policy that affects society, that's the most difficult part to tackle, and we're still doing that. And the way we've gone about it is we've had patient focus groups. We've also had to talk to policymakers, do simulations, and that's what we're doing right now. So we have genetic information, but how do we use it to guide policy? That part, I believe, is the next chapter for us.
Okay, great. Thank you very much, James. Let's continue with the conversation about bringing genomic technologies to the real world and to clinical practice. So, Bruce, while we have you, I'm hoping that Bruce is still there. Tell us a bit about where things are from a pharmacogenomic testing point of view in terms of clinical care, access to pharmacogenomics, and also use of pharmacogenomics in clinical care more generally.
Yeah, well, again, almost 1,000% increase over the time period where we started with one oncologist who said, this sounds exciting and useful. Another one said, I don't understand why you're looking at pediatric oncology. We don't have adverse drug reactions. I said, oh my God. I mean, we have them all the time. I mean, heart failure, hearing loss. Oh no, those aren't adverse reactions. Those are things I know exist and are going to happen. And so it's not, these aren't unintelligent people. They're just focused on an outcome that they believe survival has a cost associated with it, which is the loss of hearing or the worsening of cardiac function. And when they start seeing these as independent outcomes, then we have a real opportunity to improve and optimize therapy. And that's what's happened now. So we've seen this massive increase, and a lot of other areas are now asking us for testing. And I've been working with colleagues at the BC Cancer Agency to implement testing in adult patients on fluoropyrimidines. We implemented that in British Columbia. That's now the standard of care. So it's definitely improving. And in terms of the numbers of people that are tested, I do think that patients are the drivers here more than clinicians in terms of their concern. And so I want to get the test results in their hands. And that's the point of the new project that we're working on, to have inexpensive tests done, kits available in pharmacies to get more patients with information about their own unique drug response profiles that they can then use to better empower their own decision-making about medication use.
Great. Thanks very much. So maybe we'll go straight to Lisa. And you actually have had pharmacogenomic testing, and you took that to your clinician.
Yes.
So you need to press the button.
Am I on?
You're on.
Great. Thank you, Sterling. Great question. I didn't know it was coming. Or did I? I think I did. Always answer the question you're asked and don't ramble. So I'm going to say one barrier to clinical practice is the fact that there's a limited acceptance about the efficacy of PGX testing in mental health care. I've met some psychiatrists that say they don't need to use pharmacogenomic testing because they already know from speaking to a patient that they can prescribe the right drug. So there's a barrier. Of course, we're all competing for dollars, and I guess ultimately we're competing for healthcare. We all want to age well and be well, so it is a competition. So it's a resource issue. Yeah. I think delivering to every patient is also an issue because some people don't have a doctor. And if they don't have a doctor, they're going to find it really hard to access a psychiatrist. So there are issues of penetrating the boundaries. Thank you.
Great, thanks. Christian, do you want to give us your perspectives on implementation?
Sure, happy to. So I guess the best example would be the essay that I have outlined to you in my presentation, which is a gene expression-based lymphoma subtyping essay in essence. And just to state up front, this just for context, this is not at the same scale as what in other contexts we talk about, like a genome, transcriptome, some complex big data type essay. So this is a distillation, a limited readout on the transcriptome, but that has worked for us. So maybe my message here is we are working in a rather conservative system in terms of decision making and funding allocation. Maybe a bit boring is good to start with. And then just to give you the context of this gene expression-based assay, the foundation of the clinical utility and the biological validity of what we are measuring, that foundation goes back to 2000. So it is biologically a fairly old concept. Yet then the translation and implementation as we now have it as a success story is driving, it moves the needle, is clinically very relevant. So in a way, before we even started this, we knew, being in the field of lymphoma, that there is clinical utility up front. So some of the biomarkers or genomics that we're discussing are a bit vague, and there's still a gap to demonstrate the clinical utility. So that's definitely a risk. The SAFE project that I'm describing just was a convergence of, we knew already that there is a market out there. So the clinicians that we are ourselves or interact with, we know upfront that they want this. So we have created our own market upfront. And then on our end, implementing the assay has obviously risks. You have to pick the right platform. You just have to do all your validity testing and all the accuracy and what you do. You have that under control, so that's maybe the other element that has to fall in place, but you control that. You just have the clinical utility in the bag. And then the unknown, the third unknown is the uncertainty and inertia maybe in your system or in your institution or in the mindset that you have to move. That worked out for us. But to be honest, in my view, that's the most fragile. And that goes back to what we have discussed now throughout this conference. That this conservatism and that we don't know how institutions react, and that there's no top-down strategy. So if top-down everyone wants this, things would be easy. They're not, and we have to do all the things at the same time. We have to convince our leaders, we have to do the assay, and we have to create our own market. If all of that comes together, I think we have a chance, but maybe a boring approach is good to start with.
That's interesting. I mean, I think it goes back to the point that Marco was making about leadership, and actually like the importance of buy-in from leadership of the health sector, of of health organizations. I think that's absolutely right. So I do want to encourage conversation. So if people have things that they want to say, I want to go to Slido. But I also want to just like have a quick response from the panel on the equity question. Like, are there certain patient populations that stand to benefit more than others? So I think, James, you were like explicitly exploring that. There was a concern and you wanted to explore, are actually there going to be some systematic patterns where some groups of patients will be disadvantaged as a result of this? And you actually did analysis to explore that. So could you speak a little bit further to that, and then we'll get others to react, and then we'll go to questions. Thanks.
Absolutely. So there's two ways we're using the mismatched information that we have. The one way is to guide the prescription of drugs that we're delivering to patients. So the way we currently look at risk stratification is based on how many antibodies that someone makes. So a lot of women that have had pregnancies, they've been sensitized before. And so many of them are considered high risk because they have been sensitized to pregnancies before. However, when we apply the new method that we're using now by looking at molecular mismatch, they might actually very well be low risk. And so we're minimizing immunosuppressant in those patients that have been probably falsely been assigned as high risk by traditional risk factors. So that's one patient population that I feel like we've done a good service for. If we flip it around this, there are the pediatric, the young patients. A lot of them, they were assumed to have low immune risk because they've never had pregnancies, they have never had transfusions. But by happenstance, they're actually high mismatch with their donor. Now we know because we look at the molecular mismatch. So they actually would intensify the immunosuppressive medications that they get. So from a prescription perspective, I feel like we're helping people. We know better now who needs more and who needs less. But the challenge is when we go to allocate a precious organ and we're trying to match it to someone. And that's when these other factors come in, ethnicity. And I think there was a question about, well, if you are in a less ethnically diverse population like Saskatchewan, for instance, where the ethnicity is more homogeneous, and you're an indigenous person there or Asian person or a less visible a minority there, are you going to be more difficult to match? I don't know, because we haven't done our analysis. We did it in BC, and we are a little bit more diverse compared to some provinces. And so to make sure the other users in Canada, they can do the analysis, one of the goals was to diffuse the technology so that every center in Canada has the ability to perform the analysis that we did. And that was built into our LSARP. So Karen Sherwood in our laboratory is leading that effort, that all the Canadian labs have the ability to perform this technology, to validate it and do the same analysis in their own province, and to look at, are there going to be ethnic disparity introduced in this or not? And there are, then we have to figure out a solution for this. And we kind of thought that if you're in a smaller province and there's ethnic disparity, if you try to match, then we could combine several provinces together to take away that effect by increasing the donor pool size. And so you can see Western countries kind of coming together as one big geography to mitigate that. We're thinking more broadly, we can combine all of Canada to share our organs in such a way that we minimize this. So that's for down the line, yeah.
Wow, thank you. Like the thought of Canada coming together to work as a collective in this space, like just makes so much sense. So more strength to that. Bruce, anything that you want to add on equity concerns or potential populations who might be disadvantaged as a result of application of pharmacogenomics?
So, yeah, 3 quick points. The first is that the genetic diversity of genomic data is not representative of our countries. The UK Biobank is more than 60% European DNA, and the UK is not 60% European. So our genomic data banks have to echo the genetic diversity of our populations, not the populations of Canada in the 1970s, but the populations in the 2020s. And to do this, we need to think much more deeply about how to encourage all ancestries to participate and what the cultural paradigms are that are preventing that. Because it's obvious if you look at the database. I can tell you that our own data is deficient of our Chinese and other Asian brothers and sisters more than it should be. Our indigenous populations, et cetera. So that's something that we're working on, and we have a diversity task force chaired by a tribal elder and chief, also from Vancouver Island, by the way, who's a good friend of mine. And we're not just looking, though, at indigenous participation. We want all ancestries to participate and ways to do that. The second issue is that children have unfortunately been a product of poor research, and certainly, my experience is that journal editors are less interested in publishing papers in pediatrics than in adult medicine because the populations are much smaller. And I say this a lot to my trainees in the lab. Children are 25% of our population. They are a minority, but they are 100% of our future. And lastly, I would say on the equity issue, is a big problem is price. And so if government's, slow, as it has been, to fund pharmacogenetic testing, then we need other methods to move this forward. And largely the price is of testing can be affordable. The technologies are there to allow us to do that, but we need scale up. And the more scale up we have, the cheaper the individual tests become. And so that's something that I think is very important. And we need to get things down into a price point that more people can afford, and then the price can come down significantly. And that's what we're working on.
Great. Thanks very much. So I'm going to go to the first question. The most popular question on Slido is for Lisa. So we'll go to that first, and then we'll take questions from the audience. So please just raise your hand if you want to ask a question or make a comment. Lisa, your meds shows how research teams can engage with patient partners. What would you like to see funding organizations do differently to support patient partners?
I didn't know that question was coming, but that's okay because I can ramble.
Without rambling, yes.
Yes, without rambling. Well, project funding versus program funding. I think it was raised earlier by someone in the audience, program funding being, in my case, the strategy for patient-oriented research, which is a parallel to all of the institutes. So it runs on its own, but in tandem with the institutes. Some institutes espouse it, some don't. Anyways, we're on the second part of our funding. So I would say program funding, and then personal funding for patient partners. So when you have a great idea, whether or not that's driven by your patient community or by your colleagues on your research teams, you think about budgeting because you're going to one day in the future be required to have a patient partner on your team. So you start off with a budget for patient partnership. Currently, nationally, $40 an hour is what I earn. Sometimes I don't even earn that. I do a lot of volunteer work. So I would say it comes down to program funding and personal funding.
Okay.
Thank you for the good question.
Great. Thanks very much indeed. Do I see a hand? I don't see a hand. So please be thinking of your questions. So there's a great one here for Christian. So I'm going to go to that one next, even though it's not the most popular one. I think it's one that speaks to me as well. It's given the large number of tests and panels for cancer, do we have an effective system to phase out? Whoops, it's moved. Where's it gone? Sorry, yes, move to phase out the old tests and replace them with the new ones?
Very good question, and actually not so difficult for me to relate to, because indeed the the plan of at least some of the testing that we developed in LSARP always had the health economic idea in mind that it would be actually a replacement of existing cancer testing. And just to give a concrete example, fluorescence in cytohybridization or classical cytogenetics is a little bit long on the tooth. I mean, I'm offending a couple people, I'm sure, with this statement. But so that is from a health economics point of view, a good argument to make that the testing would be cost neutral in that sense, or even cost effective, or at least cost neutral. Yet this is a good argument, but in my experience, the testing costs usually in the health economic models are negligible anyway. So maybe a more general point to make, most of the modeling and driving the ICERs and all the metrics that we modeled together with Dean Regier's group came out as a drop in the bucket, whatever you do on the genomics side. And the big cost drivers are the treatments and CAR T cells and bispecifics and antibodies and so on. So what oftentimes, say, a major driver then is limiting the indication range for an expensive treatment, which is an argument that industry doesn't like. But I'm getting away from the central argument. Yes, we have plans in place that would take out the fish and swap in our new assay.
Okay, great. Lovely. Thanks very much. Okay, I'm going to stick with Slido unless I see a hand. I'm going to stick with Slido. Okay, so what about the recent failure of 23andMe? Someone was asking that and how that might actually bring about challenges to uptake of genetic or concerns about genomic data. Who wants to speak to that? Bruce and Lisa, and then others as well. Bruce, what's going to be the impact of the 23andMe failure?
Well, I think that in medical research and consented, written informed consent research, I am a steward of yours or other people's DNA, and you have the right. You never give up the right to have that, to have it back, to have it destroyed. And so I think that's one of the big differences is that we operate under that rubric. And so, and with current laboratory technology, the way it is and everything barcoded, I can find your sample or any aliquots of your sample within the lab quite easily and dispose of them in the regular biological waste. So that's not a big problem in terms of managing that. I think that these commercial companies have and be careful now, but I think sometimes they overpromise what they can deliver. And I think genetic information can be useful. I'm not sure how useful knowing that cilantro tastes like soap is if you have a genetic test, but it's interesting. That I think is one of the big issues that 23andMe has faced, is that there's waning interest in some of the testing that doesn't really serve a particularly pragmatic purpose.
I mean, I think the general point is, like, are we actually facing, and does it undermine confidence in genetic testing, period, regardless of whether it's commercial or not? Lisa?
Well, coincidentally, I can't believe it, whoever asked that question, I have a copy of an article from The Economist, March 29th, 2025. The bankruptcy of a genetic testing firm raises tricky legal questions. I mean, I don't know what happened to 23andMe ultimately. I don't know if it was sold or split up. I can't say from a corporate point of view what's happened to it. From a trust point of view, a lot of trust was shattered. And of course, that trust was shattered because of privacy concerns. So this company has data that is valuable to us as individuals, certainly to us, those of us who have suffered mental health issues, because there's stigma attached to some of that data. As an employee, you can be marginalized if you have mental health issues. So data concerns, the corporate concerns of what happens to the pieces, and the trust issues. And there's always Genomic, that terrible company in the States that's also facing corporate dissolution. So yeah, there are long-term, short-term unintended consequences, but many long-term ones.
I'm pleased that you're a lawyer when you're making comments like that, Lisa, about commercial entities in the States. But anyway, that's fine. I'm sure you're fine. James or Christian, do you want to speak to the patient's concerns around testing that's genomic? Do you experience any reservations or concerns?
Yeah, I think because of the patient population that we serve, they are getting tested for a specific reason, and that is to receive a transplant, and that testing has occurred. So the main concern that sometimes they have is about the security of the data. And so when we conduct our research, we actually we made it a mandate in our research program that the researchers have to be on site, and all the data has to be on a firewall behind the institution. None of it can leave the institution. And so we have so many collaborators, and that was our way to ensure the privacy and integrity of the data.
So it goes back to the consent, I believe, what we're saying there, and what the conversation is with the patient, and what's filed away. So our standard consent in the lymphoma group, but I'd say at BC Cancer, would obviously be an informed consent that allows broad data sharing. There's some minor nuances. Industry sharing usually is standard language. Sharing outside of cancer, some consents say it has to be cancer research, others say any research, maybe a nuance to be aware of, at least for retrospective studies. But overall, I'd like to make the point that we have to be careful to overreact. The privacy is a concern, of course, and we should safeguard this. But if you would go to 100 patients who have donated their samples to research studies, I'm just coming up with a random number, but 98 to 99% are fully on board. They think that this is one of the ways that they can meaningfully contribute, and they're enthusiastic. And I think we just have to carefully tighter the one or two, to which degree that should drive our global decision making.
Great, thanks so much. So can we go to the results of the poll? Okay.
Ooh.
Cancer detection, then pharmacogenomics, then organ transplantation. Okay, well, thank you very much for participating in that. And we are now within the last minute of the session. So I'm going to say that was great. Thank you, all of the panelists, for sharing your thoughts. And we have the results from the poll. So that concludes our session. We hope you've enjoyed the conversation. Many thanks to our speakers and to everyone for joining us online for the session, and for those people in the room, especially those who made my life so easy by asking so many questions on Slido. And I'll now invite Federica to return and share some thoughts on the day. And thank you, panelists, indeed.